И мы последний доклад, видеоролик Людмилы Зиневич, да? Так, включите, пожалуйста, ролик.
Dear friends, dear colleagues, good evening. I'm very happy to join you at this conference, even in such online format. And many thanks to the organizers for such an opportunity to present you some results of our Steppe Eagle population structure genetic study. 5 years ago, the Altai Krai, the Steppe Eagle conservation workshop, the importance of genetic research of the species was highlighted. So we're working on this problem, and we're still working on it. And now I want to show you some old data and also some new data. And we hope that it would be somehow helpful in the Steppe Eagle conservation. The key point of our study is the comparative analysis of two species, the Steppe eagle and the eastern imperial eagle, because the main problem of all conservation genetic studies is that we have no reference values of genetic diversity, of inbreeding coefficients, and so on, to make some conclusions and some conservation decisions. And so it is, in fact, recommended to compare different species with different conservation status. And so we can see for the Steppe eagle and for the eastern imperial eagle, which is only vulnerable, not endangered, and it does not show such great decline in numbers, and in some parts of area the species now is even stable. So you can see what we've done and what methods we used. I will speak about it later a little bit. And a few words about these two species and about sampling. There are two key differences between these species, which are pretty close in biological and ecological traits, and live sympatrically in some parts of areas. So one thing is that the population structure of the Steppe eagle shows clear distinguishable nesting groups or subpopulations, perhaps, which can be discriminated by GIS methods. And we tried to sample as much of these nesting groups as we can. And for the eastern imperial eagle, we took only the part of its wide area, the Russian and Kazakhstan part, and this part of the area looks like a continuous nesting group of subpopulations, just with some assemblies. It was also shown by GIS methods. And another difference is in population number, because endangered Steppe eagle still has more individuals than vulnerable eastern imperial eagle. The main thing for the Steppe eagle is that it was the most numbered Palearctic eagle species. So still we have more Steppe eagles than eastern imperial eagles. Please remember this fact, because we will need it to analyze the genetic data. So we can see from the mitochondrial DNA diversity that the haplotypic networks for both species look rather similar, but with some specific traits. First thing is that the Steppe eagle is monotypic, without any subspecies, without any haplogroups. And also the imperial eagle is monotypic, but the Spanish imperial eagle forms the separate haplogroup, so you can see how it should look if Steppe eagle has any genetically distinguishable subspecies. So the steppe eagle is monotypic, and both haplotype networks show so-called string of beads-like topology, so several widespread haplotypes. Such topology is the trace of the species, the population, coming through the bottleneck. So the period of the genetic diversity decline. And if we remember the fact about the population numbers, and if we look at the numbers of the haplotypes, we can see that the genetic diversity of the steppe eagle is lower than the genetic diversity of the eastern imperial eagle. So we can suppose that these bottlenecks in the evolutionary history for the steppe eagle were much more severe than for the eastern imperial eagle, but still the species survived. As for other genetic threats like inbreeding depression and population fragmentation, we estimated the inbreeding coefficient, and we can see that the estimations for two species are comparable. Of course, this measure is a little bit bigger, higher for the steppe eagle, but still we cannot see anything catastrophic. And as for population fragmentation, we use different methods to estimate possible fragmentation, but we saw no traces of severe population fragmentation. If we visualize this data using principal component analysis, we can see it clearly that all these separate nesting groups of subpopulations of the steppe eagle do not form any genetic clusters, geographical or not geographical, still they're not clustering. And this is rather close to the continuous subpopulation of the eastern imperial eagle. So the steppe eagle population is not fragmented at all. Next question is if this is because of the good condition of species of population, or this is because of some behavioral traits of the steppe eagle. Our very preliminary data about birds' individual returns to the nest and about the paternity testing of different birds show that from 25 nests we analyzed from the same nesting group, we found 5 pairs of closely related birds breeding the nests, different nests, the same nesting group. 4 nests were rather close to each other, from 1 kilometer to 20 kilometers or something like this, and only 1 pair of related birds had more than 300 kilometers between their nests. So, of course, it's a very small set of samples, and we cannot conclude anything, but still it looks like the steppe eagle also have natal philopatry, even some natal philopatry. So it does not differ much from the eastern imperial eagle in this trait. Even just occasionally, we found another genetic threat, the mitochondrial DNA introgression from the eastern imperial eagle to the steppe eagle population in Dauria, where both species are in decline of numbers. We found an unusual feather on the ground nest, an unusual feather with mitochondrial DNA of the eastern imperial eagle, and a clear STR profile of the hybrid, and not a first generation hybrid. As this bird turned out to be a female nesting on the ground in the steppe eagle population, then we can speak about mitochondrial genome introgression. And as any introgression, it can be a genetic threat for the steppe eagle population stability. But still, we analyzed about 400 samples, and we found only one case of such hybridization. So it looks like a rather rare event, not pretty serious threat. And the most interesting thing, perhaps, about the effective population size, we can see using the linkage disequilibrium method, which deals with nowadays population, modern population, not population history, we can see that the situation is not so bad. The effective population number for the steppe eagle is rather high, and of course, a little bit higher than for the eastern imperial eagle. But the interesting thing is that molecular ancestry method, which deals with the last bottleneck, with the last period of decline of the genetic diversity, again shows us the trace of very severe genetic diversity decline for the steppe eagle, much more than for the eastern imperial eagle. Same thing we saw using the mitochondrial DNA markers. So we can conclude that such population number decline and genetic diversity decline, as we see nowadays for the steppe eagle, are not new in the evolutionary history of this species, and this species survived much more severe declines. So we can hope that that's nothing catastrophic. And we found no critical genetic threats for the steppe eagle for nowadays. The population is genetically monotypic and not fragmented, still not fragmented. And as they show some traits of natal phyllopaty, perhaps specific conservation measures at key nesting territories can help saving the genophond of the species and save the species from the extinction. And of course, we should monitor genetic diversity, effective population size, and inbreeding coefficients, and fragmentation, and all other possible genetic threats. And also, we can recommend to monitor somehow the hybridization with the eastern imperial eagle, because it can be a threat for the steppe eagle genome uniqueness and population stability. But again, this looks like, nowadays it looks a rare event. And so perhaps as a conclusion, perhaps the steppe eagle is not only a bird of prey, but also a kind of genetically, a kind of, I don't know, a lemming, flying lemming. So we can, perhaps we can hope to save this bird and to see the population number restoration, because this bird saw such periods in its evolutionary history. Thank you for your attention.